Okay, so this is an update on the current state of the Digital Botanical Gardens Initiative. You can find the slides here at this Zenodo address. And there are a number of people involved, and here we only listed those that were directly involved in this presentation, but the consortium as such is larger. Yeah, background, the basic idea is using botanical gardens as platform for ecological research, specifically with a metabolomic angle. And for that, it's important to know that the Digital Botanical Garden Initiative has a, let's say, a larger perspective. So we want to build the Earth Metabolome. And for that, there's the Earth Metabolome Initiative, and the Digital Botanical Garden Initiative is kind of a prototype for this. With one key thing to consider here is that in field work, we have different parameters that affect the way you can do research. Actually, I'm affected by the Zoom stuff here. And I'm trying to hide this. Okay, yeah. And we want to build mechanisms that allow us to study metabolism in both field conditions and in lab conditions and anything in between, essentially. In terms of what can be there in between, some other things, or basically we need a balance between the hotspots of biodiversity and the hotspots of analytical capacity. And for certain purposes, you could go either way, or you could choose a middle ground. And actually, botanical gardens are somewhat of a local maximum of both biodiversity and analytical capacity, and that's what makes them interesting. So we shuffled things around a little bit, and adding a digital dimension to this is also an interesting component here, so that we can, in principle, interact with all these different ways of sampling ecological interactions, and we can bring them together and make use of information from multiple sources. So the basic idea here is to establish a robust workflow for gathering metabolomic data. Then we prototype this in botanical gardens in Switzerland. We automate to the extent possible, and then we scale things up in multiple ways and branch out from there. We do have a plan, but we don't have time to go into too much detail. So I basically give you some indicators. You are here in the botanical gardens. I'm mostly here on that corner as a data scientist and involved in these kind of projects. There are many ways for you to get to me or for me to get to you, and a number of other ways to move around in this system. I put the magnifying glasses here on some of the things I will briefly touch upon, but the system is much more complicated than that. Okay, so one thing, for instance, is how do we collect things? And here we go from specimen to a sample with taxonomy and also geolocation. So we make use of a crowdsourcing platform, iNaturalist, and we, this results in some physical specimen and also a digital specimen represented in databases. Then from the physical specimen, we want to extract information. So some additional steps come in, and we keep track of the specimen itself and metadata about what has been done on it already, and also all the information extracted from it. This means that we need to have mechanisms to connect the physical objects across the entire workflow. And these mechanisms, they need to work in a way that is essentially digital, but works in the field and in the lab. And yeah, then we want to combine this with data analysis, particularly mass spectroscopy. And that, again, has a number of individual steps that we need to go into. And yeah, then how can you find out about the current state? One way is to go to iNaturalist, essentially, where we have a dedicated community, and where you can see how many observations we've made, how many species are involved, how many people are involved, and things like this. Here's a typical entry. So you have the specimen, basically the sample in its tube with the taxonomy and the original plant from which it was taken. You have the geographic information and some additional tagging. Plus you have the option for community interactions. And while the focus is on plants and flora, and we do have some fauna examples as well. And for the Earth Metabolome Initiative, we do think broadly, but we start out with the botanical gardens. In terms of coverage, the focus right now is mostly on Swiss botanical gardens, and here is the two main ones that we're working with, Fribourg and Neuchâtel. And so the total number of samples is several thousand, and they're in different stages of the process, and all of this keeps growing and is developing. While we're doing this, we keep an eye on the things that can be done towards this more global goal of the Earth Metabolome Initiative. And so we're testing our methods in a small scale of the individual botanical gardens we already have, the species we already have, the kind of sample techniques, also sample labeling techniques that we already have. And then we do some testing and prototyping around that. For instance, we did a BioBlitz, well, several actually. Here's a one given as an example. And the idea here is to gather a team that does not only include the core team from the project, but also people from a bit more outside, so that we can gather data of the kind that we're interested in at a larger scale than what is normally possible in a botanical garden. Also, we can gather it in more field conditions, and we can involve people of different, let's say, familiarity with our workflows. Because in the end, if we want to scale this globally, we need to involve many more people, and we have to make it easy for them to engage. Okay, how can you get involved? There are various ways. One, you can review what we've done already. Then you can consider whether you're doing anything that goes into the direction of metabolomics or metabolic studies, whether you're doing anything at scale with multiple species, multiple samples, multiple sites, multiple people, time periods, methods, anything like that. Or if you're working with knowledge graphs, because we're building a knowledge graph out of all of these kinds of information. Or if you're doing anything that zooms in on metabolic aspects of some biotic interactions or ecological interactions. These would be things that we could also be interested in collaborating on. And then, so if you have things like that, then we can maybe think together how this would fit into the DBGI or EMI initiatives. And if not, but you would like to get involved, well, then we can discuss how some of the things we have might be relevant to you, or some of the things we would like to have might be relevant to you. And so basically, this is a pledge for you to consider DBGI and EMI when you're doing the stuff that you're doing anyway, and especially if you're making new plans, and also if you're involved with teaching. Because you can use our resources for teaching about aspects of metabolomics and phenology and things like that, but also data science. Yeah, with that, I'm providing here contact. We have websites for both projects. We have emails. We're on social media. We have a community on Zenodo where you can find a number of additional slides and data sets and things like this. And these slides, again, are on Zenodo as well. And some of my colleagues are also on the call. And so together, I hope that we can address any questions you might have.